Okay, we start the lesson from the last week, cell cycle and the process of cell division. We briefly look cell division. We will see what exactly we have to remember for the exam, right? Okay, I try to draw it again. Here. So I said the cell cycle is the events which take place from division of one cell to two cells. I mean, one cell divides to make two cells, right? Like this. If this is one cell and this cell divide, and we can see two cells, okay, like this. So we are going to learn what are the things happen from here to here. So we divide cell cycle into four, if not maybe five, right? So five stages or phases, right? Stages or phases. First one is it's like a G stand for growth. G stand for growth. Growth one, first stage of growth. Then the next one is S stage. S stand for synthesis. G stand for growth. Then after that, here. Growth 2, second stage of growth. Then this one indicate for mitosis, that means real division. This also include division of cytoplasm, which is usually right in C letter. Right? I write here cytokinesis and M mitosis. Right? So these are the stages of cell cycle. Usually, the mitosis is the division of the nucleus to make two cells. So this is usually very short, 10% of the total duration. For example, if this is 24 hours, the cell cycle duration is 24 hours, 10% is 2.4. 2.4 hours is the mitosis. And I said also G1, S, and G2. So these three stages collectively called interphase. The time taken for G1, S, and G2 is the interphase. Right? All right, so this is important to remember now. If you can draw cell cycle in a paper, and then you can roughly allocate the time for each one like this, and mitosis is just 10% of that. Right? Okay, now if we take this G1, S, and G2, we need to remember what is exactly happening in each stages, right? Let's name what is exactly happening, right? Now, growth is cell growth. It means simply the cell can increase in size. How it increase in size? It can increase the organelles, right? Number of organelles increased. Number of organelles increase. Another important thing we need to remember, if we take what is the most important biological molecule in a cell, or what is the important biological molecule in human body, we have learned carbohydrate, protein, lipid, what else? DNA, water. So these main biological molecules we have learned: carbohydrate, lipid, proteins, DNA, water. Out of all, the important functional molecule, functionally, what is the most important molecule is the protein. Because protein can do a lot of things inside the cells. It's like a structural and functional. It can make a structure and it can also do functions. Because if we consider other molecules, like carbohydrate is an energy molecule, lipid is energy and structural molecules, and the water is like a base for everything. DNA molecule is the control molecule of everything. So it's a DNA and protein are the two important molecules in a cell. So DNA control everything by making protein. So we call it protein synthesis. Right? Now you can remember G1 stage, number of organelles in increase and protein synthesis take place. So S phase. S phase, I explained about the DNA molecule. Briefly, DNA molecule make one DNA molecule makes two DNA molecule, right? So this is called synthesis of DNA, right? We write here synthesis of DNA. This is also called DNA replication. Simply DNA doubled. Then the G2 phase, it is again synthesis of protein. Now, if you want to understand, the protein is required for this S phase. Most of the proteins needed for the S phase. And synthesis of proteins, maybe this protein is needed for the M phase. Example, for the M phase. So they have to make the proteins. M phase need the proteins, so they make the proteins. And also, when we talk about the organelles, we learn a lot of organelles like mitochondria, RER, SCR, Golgi, nucleus, the lysosome, peroxisome, and ribosome, and centromere-like things. Here in G2 phase, there's one important thing happening, but it's not in the resource book. You know, sometimes very important things, even it's not in the resource book, we need to learn, right? Why is that? Because resource book is made by someone, they might miss certain things when they make this resource book, and it's not revised. So far, we have the new syllabus book, and the book is not it's not like a going as a standard textbook. Like textbook are changing, and then have editions, edition 1, edition 2, but that is not really textbook. So these kind of things are not really happening, so they miss certain things. One important thing is the centriole. So centriole, how we see centriole, we have seen they exist in pairs, right? They exist in pairs. One centriole perpendicularly oriented to the other centriole. So it's called, together the pair of centrioles are called centrisome, right? Centrisome duplicated. That's doubled. Centrosome duplicated or doubled. That's very important. We will see why it is important. The central duplication is important, right? Then after that, M pace. Even this is 10% of the time for total duration. We need to learn more on mitosis and meiosis, the other process. So we don't learn these G1, S and G2 in details, but we try to remember what is exactly happening in these stages. Right. So synthesis, S, G growth, and M for mitosis, C for cytokinesis. So this we're going to learn in details. So no need to remember anything at the moment here, but here number of organelles increase, protein synthesis take place, and synthesis of DNA, and this is also synthesis of protein, and also number of organelle increase. Organelle increase, example, centrosome duplicated. And then G1S and G2 is known as an interphase, which is the longest stage. Right? Okay, so I will just mention in the next coming up what is exactly happening, and we proceed to the G1. Here again, we have a G1, then S, G2, and M, and cytokinesis. So if we take G1, what is exactly happening in G1? One is protein synthesis and the growth of the cells, and then growth, organelles increase, and protein synthesis. So we can list only these three things happening in G1 phase. Then S phase, DNA replication. Okay, here I mentioned last week, DNA molecule is like this. And I clearly mentioned it is very difficult process because DNA molecule, person to person, we can see some changes. Where we can see changes, we can see changes like this. Let's say this region, if we take this region, we have a order of nucleic acid like adenine, cytosine, guanine, thymine, adenine like this. We can find a special order. Then this order exactly copied to the other molecule. That means when it makes two DNA molecules, this order ACCGTA should be exactly in this molecule and this molecule, and they should be the same. And the difficulty is roughly around 3 billion nucleotides in one strand. We call this is one strand. Two strands make the DNA molecule. Two strands make the DNA molecule. So now 3 billion in one side and 3 billion in the other side. We will see DNA molecule, how it copied in details in molecular biology. Just briefly to understand how it happened, this DNA molecule, the two strands separate, right? Two strands separate mean, I just show a part of that. Now you see the two strands separate. When the two strands separate, we can find the nucleotide order. Right? let's say here if it is A, here C, C, G, T, A. I take the same order, A, C, C, G, T, A. And what is we can find in the other side, if it is A, then this should be T. If it is C, this should be G. If it is C, this should be G. If it is G, C, T, A, T. So we have learned in DNA molecule, this is called Watson and Crick complementary base pairing. A always pair up with T, C pair up with G. Now, if we want to make a new molecule, so when you want to make new molecule, so this in front of this, the new molecule is formed like this. So here, if it is A, this should be A, this should be T, and this should be G, this should be C, this should be C, this should be A. Right? Now this one become one molecule, and this side also happen in the same way. And this become another molecule. After that, this make a one DNA molecule, and this make one DNA molecule. We will learn the process in detail, but just to understand how it happened, DNA strand separates, and each serve as a template. The yellow color one is a template. On top, on that, the white one, the new strand is made. Right, so that's a very important process, and we have to also understand if there's a mistake happen. So mistake happen when it trying to pair up, mistake can happen. Or before that, it is nothing to do with cell cycle. Let's say a person DNA, we call it DNA damage. DNA damage. Now, what is DNA damage? Due to some kind of chemicals or maybe exposure to the harmful rays like UV radiations, X-ray radiation, some chemicals, they can damage DNA molecules. So if the DNA molecule damage, let's say certain nucleotide are missing and they are no longer there in this DNA molecule. That means DNA is not the correct DNA. So that can happen due to external factors. And internal factors, when it make a new DNA molecule, there could be a mistake. That means DNA damage, it's not the correct DNA. So this can happen in S space, or maybe already DNA is damaged in the S space. That means when it copied, it can make a mistake. But before that, due to some chemical, DNA can be damaged. We'll see why it is important for us to learn in the last section, that is end of cell cycle and mitosis and meiosis. There's a small topic called cancer. So it is important to understand the basic concept if we want to understand about the cancer. We have some idea like cancer is a common thing, and we have a small idea what is cancer. But keep in mind, DNA molecule can damage due to external factors, or DNA molecule not correct order. This is not in the correct order due to a replication or synthesis problem. So next, we come into the G2 phase. What is G2 phase? Important again is the growth. Organelle increase, and I mentioned centrosome duplicated, and also protein synthesis. So growth, organelle increases, centrosome duplicated, that is additional things which is not in the resource book, but that's important, and protein synthesis. Then M-phase is the division of the nucleus. Now, we have two types of nuclear division. One is mitosis, other one is meiosis. So we're going to learn in detail mitosis and meiosis. So what is the basic difference of mitosis and meiosis is, the basic difference is, if we take a human DNA, the DNA molecules, it's a long molecule, as I mentioned. These long molecules breaks into small divisions called chromosomes. Chromosome is the organization of DNA molecule together with the proteins, and the chromosome number in human cell is 46. So what is it? 46 molecules of DNA we can find in human cells, and the 46 exist in 23 pairs. So this number 46 or 23 pairs is called diploid. What is diploid? It's a full set of chromosomes. In human, 46. Right? We just abbreviated by 2N. Diploid is the full set of chromosomes. If you take a human, it is 46. If you take another species, it's different. Chromosome number is different from species to species. So 46 is the diploid number. Okay, what's the difference in meiosis? Meiosis has half of the chromosome. Meiosis has half of the chromosomes, so it is 23 then. It is 23 chromosomes, and this is called haploid. So meiosis produce a cell nucleus with a half a number of chromosomes. But there are other differences. We're going to see these differences in mitosis and meiosis. But generally, mitosis is cell make another cell which has 46 chromosomes, and the cell is exactly same. Meiosis, it make a half number of chromosomes. Then next stage is the cytokinesis. So what is cytokinesis? Cytokinesis is the division of cytoplasm. Okay, we also learned what is interphase. Interphase is the combination of G1, S, and G2 phase. That's called interphase. And that's quite long. 90% of the time taken to go through this G1, and G2 phase, right? Okay, next, we have this checkpoint. Now, what is checkpoint and why it is important? And where we can find checkpoint, we have to look. And also, we will look a little bit more what is this checkpoint for understanding purpose. Here is a G1, S, G2, and then M, right? So don't worry about too much always the time taken for these microbes is not always the proportional to the time taken to complete this page, right? Okay, first, where is this checkpoint and what is checkpoint? We can find one checkpoint here. And we can find one checkpoint here and one checkpoint here. That means we have a G2 stage, S and G1, right? Or maybe between G2 and M here, this is S and G2 stage, and G1 and S. Between G1 and S, and then S and G2 and M. Why this checkpoint? Now you see, end of G1, it should proceed to S. End of DNA replication, it should proceed to G2. After G2, it should continue to M. Once M start, once it enter the M stage, we cannot stop. That means there's no way to stop cell division. But cell division can be stopped at G1 stage to S. Cell division can stop at S to G2. And cell division can stop G2 to M. But M start, if it goes to the M stage, it's very difficult to control. That means we can have a two signal. One is at each checkpoint, we can give signal go ahead or stop. That is the two options, either continue cell division or stop. Right. Why it is important to check the cell division? Because let's say the DNA is damaged. The DNA is damaged, and if the cell make another cell, two cells, the cells are wrong. So it can make two cells and their DNA is not correct. Right? Let's say S stage, DNA replicate. So when DNA replicate, or DNA make two molecules, the two molecules may be wrong. It make a mistake. So the fault, a wrong DNA is going to the G2 stage. So they have to control the cell cycle at each checkpoint. So checkpoint is a control point of cell cycle. So we can find this at one phase to another phase. I mean G1, G2 and M stage, we can find the cell cycle, right? So here, here, and here. These three stages, when it going to the next one, we can find the control system. Okay, now the question is, who is controlling? It's a little bit of weird because cell is busy. So they are completely busy of doing something, and they have to also check the process. But nothing is coming from externally. The control system is also coming from the within the cell. The control checkpoints, everything inside the cells. I think this is the knowledge you need in terms of exam point of view and in terms of our reference materials, that mean the resource book. But you know, sometimes if we need better understanding, we need a little bit of extra knowledge just for the understanding purpose. If it is not clearly understand, it is difficult to remember. If we understand clearly, it is easy to remember. Because if it is say there is a checkpoint, they control cell cycle. And then you also remember there's a checkpoint to control the cell cycle. But when it comes to a little bit more details, some like if you scroll down, there is something called cancer. And how cancer happens. Then it's again a little bit of questions. It mentions certain things, but then it's very difficult to understand, right? So here, what you need to remember, G1, G2 and M stage, when it going to the other stage, there is a control points. What it control basically, the DNA damage, whether DNA is correct or not. And mostly, it's about the DNA molecules. There could be other things, but main molecule is the DNA molecules. Right? Okay, to get a little bit of extra knowledge, we have to join again for the next session. Okay, now we have a completely new phase, G0 phase. So we haven't seen in the cell cycle this G0 phase. So what is this G0 phase?